Right. Hello, my name is Billy Caruso, and with the help of Matthew Gakura and Gary Vanderlyn, I created this presentation and about my research on an mRNA vaccine design to neutralize African swine fever viral infections in pig populations. So to start out with, I think it's super important to understand what African swine fever is. So African swine fever is a virus that is caused by swine fever virus. It is a linear double-stranded DNA virus and it is between 170 and 192 killian base pairs. And then it has no effect in human. So if humans are exposed to this virus, nothing will happen to them. They will not be infected even slightly, but it does affect pig populations. So that is going to be Suscrofa. And in these pig populations, the mortality rate nears 100%, which means it's incredibly lethal and has a big impact on the pig population, which is a very vital protein source. So it causes a lot of food insecurity, which I'll be talking about a little bit later. And it manifests as a hemorrhagic fever, and it has a bunch of different signs and symptoms that includes fever, chronic or changes in appetite, vomiting, nausea, weakness, and skin rashes. So those are how you can physically tell if the pigs are infected, and then it can present in a couple of different ways. So that includes chronic, acute, subacute, hyperacute, subclinical, and inapparent. So those all present a little bit differently, but they're all different ways of how the virus shows itself in different pigs. So then a little bit more information to the epidemiology behind it. That's going to, so it is a vector-borne arbovirus, and it is also spread by formites. So that includes shoes, clothes, and then water and food. So for example, a really big way that African swine fever is spread is if one farm has an outbreak, the individual moving from one farm to another has some of the viral particles on their shoe, they bring it into a different farm, then that farm then gets infected by African swine fever virus. And then there's also a couple different carriers. So that's going to be the brush pig and the warthog. So to combat this in a lot of different places, they have made physical barriers and walls that they built to keep out these wild animals from infecting their livestock to help reduce the impact of African swine fever virus. And it does oftentimes occur in cyclical outbreaks. So at the same time of year, it does continue to outbreak and it goes away and then it gets worse. So that is something that you have to be aware of. And then government interventions. So because this is such a big problem for veterinary practices as well as food practices, the government has put some innovations in place. So that includes you're supposed to report it, and then all animals infected must be culled, which means that they get killed, or quarantined, which leads to the lack of the spread of the disease. However, this is really bad for profits. So there's also a bunch of different measures that are supposed to be put in place to combat the spread. So it's like a lot of different cleaning and getting rid of different products, and that's just not very economically affordable, especially in these third world countries. So it does often get ignored, and these farmers aren't listening to the proper or doing the proper steps to follow the quarantine measures. So it does lead to an increase in the spread of African swine fever in these pig populations. So because of this, you would think, oh, it's very lethal, there should be a treatment, but currently there is no treatment for African swine fever virus, just culling and quarantining, which isn't the most effective. And there's no vaccine as of right now. So with the help of Gary and Matt, we did design a potential tetravalent vaccine to combat 4 different structural proteins that are vital for African swine fever virus. So that is going to be protein 12, protein 30, protein 52, and protein 72. These are all structural proteins that are essential for viral entry. So how we came up with this design, we found these 4 different viral proteins, and then we were able to find the sequences. And then after we got the sequences, we got all the different strands. We did a cluster omega on JALView to align them and find the consensus sequence. And then once those consensus sequence were found, we were able to run them through a BLAST-P to check for pig anaphylaxis with already present pig proteins. After that, we were able to reverse transcribe these regions to make them into the correct template form. And then after that, they were added to the Moderna template for the COVID-19 vaccine and loaded onto that with the specific immunogens on them. As you can see, we added the 5' UTR, a start codon, the specific immunogen open reading frame from the specific viral proteins, a stop codon, a 3' UTR, and then a polyadenosine tail. And so that's how we determined how to make the vaccine and then how that's going to work. So if a specific farm or any animals, they do get vaccinated, that is going to help have the pig have immunity against this virus. So, using its MHC receptors that are present in the immune system, be able to create B cells and memory B cells to create antibodies, to be able to make plasma cells in order to fight off this disease so that it will not be 100% mortality rate and that these pigs will be able to survive. And then upon a section exposure, so 2 doses of a vaccine, it will be able to have an increased antigen antibody response so that it will better be able to fight the virus when it is exposed to it in like the real world so that it will not suffer from the disease and that will lead to immunity of the pigs. So this is super important because of the food insecurity problem that is caused by African swine fever. So a lot of different countries rely on pork as a protein source, and it's normally relatively cheap. But because of this, it has huge economic impacts. When there's a decrease in the supply, that means the demand is going to go higher, and that means prices are going to go higher. And a lot of places can't afford that. As well as there was an outbreak of African swine fever in China in 2018. And China is the #1 producer and consumer of pork in the world. So then because of that, they had to resort to different protein sources and there was a 1% fall in GDP and also the caloric intake per person decreased because it was such a hard hit to the pork industry. And one of the major implications of that was that there was an increase in wet market sales. So that includes different protein sources that were then utilized, which includes fats that harbored SARS-CoV-19 virions, which then were able to be introduced into the human reservoir. So by maintaining these pigs and these other this protein as a protein source, it will reduce the likelihood of other zoonotic diseases to have the potential to infect the human reservoir and make us vulnerable to a disease just like COVID-19 happened in the 2019 pandemic and the ongoing pandemic. And we all know what that has been like. So yes, that is everything about African swine fever and the mRNA vaccine design that we have made to combat African swine fever. Thank you.